I don't want to take up. Great. So I'm on one of the co-chairs of the seminar committee, so I'll introduce myself. But I think I know most people here. So today, as you'll notice, there is a camera in the room. So if you
Katrina,
don't,
can we get people sitting in all the seats around here? And please feel free to sit.
Are those seats more valuable than this seats?
Yeah. Okay. Maybe you can wait till everyone has their lunch. Okay. Yeah. As you notice, there's a film crew here, and I won an award. So they are from NSF and filming me for the past 2 days. But if you don't feel comfortable being in pictures, feel free to sit in the back. So I'll be doing a presentation on giving an overview of my research, and then they might interrupt me while I'm talking to move in certain locations. So, but everything I'm presenting is real science. So. But thank you everyone for coming. Today I'll talk about my lab and a couple of the projects that I'm currently, and my lab is currently working on. But today I'll mostly focus on the nicotine metabolism, using nicotine metabolism to tailor smoking cessation treatment in American Indian and Alaska Native peoples. Okay, so I thought I'd start off with an overview of my lab. So I do both research in pharmacogenomics as well as in bioethical research. So on the top, I study primarily the cytochrome P450 enzymes and work with tribal communities. So one of the projects is related to nicotine metabolism and how that relates to personalizing medicine. So I've been working with the Strong Heart study, and in the past I've worked with the South Central Foundation, and then right now we're working with the Confederated Salish and Kootenai tribe on projects related to smoking cessation and nicotine metabolism. The other aspect of my research, number 2, is cytochrome P450 diversity. So right now I'm working with a couple of tribal partners, one the Metlakatla tribe in British Columbia, where we are looking at overall diversity in CYP P450 enzymes. And then with a project with a collaborator in South Korea, we're looking at pharmacogenomic variation in the 1,000 genomes. And then more recently, my background is in evolutionary genomics. So a lot of my work as a PhD student was really looking at the evolution of how what genetic variation we see in our populations today, how did they come to be and why? So I looked at a lot of natural selection as a graduate student. And so right now I'm really interested in looking more at pharmacogenomic evolution and why we see the variation we do today. So that's a new aspect. And then 1, 2, and 3 are sort of all looking at variation in populations and individuals, but that variation we can do in silico sequencing or in silico prediction of what those variants do, but until we can actually functionally verify, I think that's what the 4th aim is, to using multiplex assays to assess what these variants do. And we have a collaboration with the University of Washington, Dr. Doug Fowler's lab and Maitreya Dunham, who are using cell culture assays to introduce variants, and these are high throughput assays. So our first assay that we're doing is on CYP2A6, which is one of the enzymes that I'm really interested in. But with this high throughput assay, we can introduce a single point mutation at every single site in the coding region, and then assess the abundance, and then eventually assess expression. So that's something that we're still working on in the wet lab. Hopefully our cell culture does not get contaminated again. And then, as I mentioned, with a lot of my work with tribal communities, in order to start to do genetic research, there has to be a lot of conversation, and tribal communities have to feel comfortable working with you. And so the other aspect that has evolved in my research is thinking about the cultural and bioethical issues. So one of the big projects that I'm leading is with the Navajo Nation, which is my own tribal community. But the Navajo Nation has had a moratorium or a ban on genetic research for the past 22 years, right? For 22 years. And the ban essentially allows no genetic research to be conducted on Navajo citizens on the nation. So, and of course, people off the Navajo Nation can participate in research, but it's also very limiting for people, urban Native Americans. Since we know there's a ban, we don't feel comfortable participating in research. So that is a project that I've been working on for many years with the tribal community, and we have done interviews, we have done focus groups, we have done surveys with the community. So that is a big aspect of what the work I do. And then the other aspects, so I do some work with tribal communities on thinking about ancient DNA and ancestor DNA and what are the ethics behind using those samples in research. And then lastly, so I run a couple, or I am a part of a couple of training programs for Indigenous students. One is the summer internship for Indigenous peoples in genomics called SING, and the other is called the PRIME program. So I'm sure everyone has seen our PRIME speakers. We have one more slated for May, so as a part of that program, we also have an early career faculty program for Indigenous scholars. So today I wanted to really talk about two projects in particular. One, the work that we're doing with the Strong Heart study, and the other, the work that recently got published looking at archaic genomes in Neanderthals and Denisovans. So I think everyone may know this, but there are many archaic elements that exist in the human genome. And this is just a map showing your percentage of archaic DNA. So if you, depending on your background, you could predict about how much percent. So it's anywhere from less than 0.1% to 1.6% in populations. And this is quite old. So actually they're able to predict a lot more percentages in other populations. But one of the pharmacogenes that I study are involved in absorption, distribution, metabolism, and excretion processes in the body. So these CYP450 enzymes metabolize hundreds of xenobiotic compounds, so compounds found in the environment and also within our own body. So the CYP gene family includes 54 genes. 11 of these enzymes metabolize actually 75% of our the drugs that we intake. So these 11 genes are the ones that we wanted to study. And a lot of this might be tied to interactions with our environment, and I'll explain that a little bit more. We know that our ancestors all evolved in the locations where they stayed for a long time. And in these locations, there are plants and other things in our that are important for our environment. And plants are rich in many different compounds, some of them which might be very uncomfortable. For example, there are hot peppers. These were natural defenses that plants have evolved to stop being eaten and to be able to fertilize or introduce progeny in the next generation, but we still eat them because some people like spicy food. But there are others that people are pretty familiar with, like poison ivy, poison oak, and then some other toxic chemicals that we like to intake, even though they're not so great for us, so caffeine and ethanol. But in this project, we know that modern humans evolved in Africa with their environmental, and then we spread across the globe, being exposed to new environments and different things that might be important for us. What we know from recent studies is that, this was from 2016, but many studies show examples of local human adaptations. So these are just different phenotypes, so like lactase persistence, height, Arctic environment, that there is evidence that these have been adapted to their local environment. A really good example is also altitude. I think that's on here. But these are variants that have been positively selected in specific human populations. And we know that when we think about archaic variants, Neanderthals and Denisovans, that the human genome is generally depleted of these regions, but there are specific regions shown here. In red are Neanderthal genes, in blue are the Denisovan genes, but these regions have been positively selected, and some might be people might be familiar with, like the EPAS1 helps Tibetans survive in low oxygen environments. So with this paper that we recently published, this is in collaboration with Fernando Villana at the CU Boulder, but we were able to look at 3 Neanderthal full genome sequences as well as 1 Denisovan individual, and we targeted these 11 genes that were involved in about 75% of the metabolism of medications, but we were able to predict exactly the diplotypes of different individuals. So you're showing the Denisovan on the left side, and then 3 Neanderthal individuals on the right, and we've color coded them to show poor metabolizers in red, and then going all the way to normal in green, and then ultra rapid metabolizers in blue. But this is the variation we see, so it's really interesting that in the Neanderthal individuals for CYP2B6, we saw star 22 in the 2B6 gene, which makes these individuals ultra rapid metabolizers. And then for the CYP2C19 in the Neanderthal individuals, we see a star 8 genotype. So this indicates these individuals may have been poor metabolizers. We were also able to look at copy number variation, and it was really neat to see that some of the copy number variation we see in populations today, we also see in Neanderthal individuals. So we're not sure if these are shared, or well, they're shared, but we're not sure if we got those, or if this occurred separately, or it's because of the shared or interbreeding with modern humans. So just a snippet. So our next steps are to examine all 57 P450 enzymes, and then also to look at more genes involved in the ADME process. Actually, out of this result, CYP2B6 is actually really interesting. We found that there was a really interesting haplotype that was only found in certain human populations in Africa, and then this haplotype was shared with Neanderthal individuals, but only in these 10 individuals from Africa. So it would be really interesting to look at that enzyme a little more closely, and then eventually it would be great to do an environmental wide association study with some of the publicly available genome data, as well as looking at different environmental variables. And then also one of the other projects with the Metlakatla tribe that I'm conducting is looking at ancestors and contemporary Indigenous populations to see how these allele frequencies have changed over time, and if there is any adaptations that we can see that have occurred in that population. So I'm going to switch gears into some of my other work looking at nicotine and tobacco cessation. It's known that American Indians and Alaska Natives have higher prevalence of smoking compared to other racial groups. But there's also a cultural connection for tobacco for many tribal communities. So it's important to remember that cultural connection. Okay. So one of the enzymes that I study is CYP2A6. This enzyme primarily metabolizes nicotine. But we're able to measure this biomarker nicotine metabolite ratio in plasma samples, urine, as well as saliva samples. So this really gives us, if you're a smoker, we can measure this and get a sense of what your nicotine metabolism looks like. And the NMR, we call it, is a biomarker for CYP2A6 activity, and also there are some clinical outcomes that are related. So, for instance, if you have a lower NMR, that indicates that you have lower CYP2A6 activity, which means you have slower metabolism of nicotine. And then, actually, clinically, there's a greater likelihood of you being able to stop smoking. But on the other hand, if you're a rapid metabolizer, it's much harder for you to stop smoking. And also some other clinical outcomes, COPD is much higher in those faster metabolizers, as well as a lot of comorbidities related to smoking. So in Alaska Native and American Indian communities, and I'm showing in this red box that NMR is actually much higher in those populations than other populations. So this is compared to white and African American populations. With my work with the South Central Foundation, we found many unique variants and novel variation in the CYP2A6 gene that actually indicates or gives us some idea of why we see such high NMR values. So one of the studies that we're working with is the Strong Heart study. So the Strong Heart study is one of the longest epidemiological studies with American Indian communities. So it includes 12 American Indian tribes across the United States. And in previous work, it was found that tobacco use was found to increase cardiovascular disease and other comorbidities in the Strong Heart study. These tribes are from Arizona, Oklahoma, as well as North and South Dakota. Right now, the Strong Heart study is in phase 7, I believe, but we were able to get data from one of the original cohorts, which was back in, it started in 1998, but we were only able to get samples from the phase 2, which is 1993 to 1995, so over 30 years ago. So it was really amazing that we could get these samples, but what's really good about this data set is they have genome-wide mega chip data as well as methylation data. The only thing our lab had to do was measure nicotine metabolite ratios, which is, and with this method that we adapted, we were able to create a protocol in our lab where we were able to accurately measure these really old samples. So we have 790 smoker samples and then a couple of control sets. For the demographics, this is about half, or see the percentage, 433 were female, and then the age range was actually 58 years old, and then mean cigarette was 11 cigarettes per day for this population, and that's actually pretty low compared to other groups in the United States. And then looking at smokers at collection, these are from the 3 different sites. Certain sites had a lot more samples than others. So Arizona, we only were able to get 35 individuals, but this is just stratifying between male and females from the Dakotas and Oklahoma. And then looking at cigarettes per day as well. So we have anywhere from very low levels under 20 to 60 cigarettes per day, which is pretty high. So once we got these plasma samples, we were able to extract nicotine metabolites in the lab and measure them using liquid chromatography mass spectrometry with the School of Pharmacy here. So a lot of this work was Crystal and my other research assistant, Kaya. They really did a great job creating this whole assay to work in the lab. And then this is some of our preliminary data from some of the NMR data, but I'm showing the mean is about 0.44, and this has been updated since I presented this slide. But when we compare it across other studies that look at Native American and then European American, it falls in line. It's a bit higher, which is what we would expect. And when we separate these by sites, we definitely see higher NMR in certain sites than others. And then it's known that NMR differences do change based on sex, and that's what we see. So we have female and male, and so it's a little bit higher in females for this group. But we're excited to finally have the genome-wide data and methylation data. So we will be conducting a GWAS and a EWAS in the near future. So some of our summary is that CYP2A6 plays a major role in nicotine metabolism, and then there's
I'll see you at 5 o'clock, okay?
Yeah, thank you. There's potentially novel genetic and epigenetic influences on NMR. So I'm really excited to continue this research with the Strong Heart study. Actually talking with their community group, they are actually really interested in doing a smoking cessation intervention with the different sites. So I haven't run an intervention study before, but I'm really excited to learn more because there have been really novel interventions that have been done in other communities, such as maybe using NMR to guide smoking cessation. So we could do that by giving a certain group rapid metabolizers varenicline rather than the nicotine replacement patches, or if we wanted to create something like a digital app, other places have made more apps culturally appropriate for the communities. So I'm really excited to continue to work with them. But all of this sort of brings together my research program in which I'm not only looking at the genetics of CYP enzymes, but also thinking about the bigger picture. And this was recently covered in a paper that my colleagues and I published, but I really like this figure because we are trying to create a holistic approach to bettering health and well-being in Native communities, and we're really advocating for a community-based approach. So the way I read this figure is that first, in the center, we have to engage all of the stakeholders, with the community being the most important stakeholder in research. And then we can increase knowledge about pharmacogenomics, so doing more genotype-phenotype association, including more Indigenous groups, looking at novel pharmacogenomic variation. And then that's only the first step. The next step would be trying to implement this in the community. So really thinking about how the work that a lot of the CCBM Biobank does and genetic testing, and how to return those results, interventions, and then really in the last part, really trying to bridge the traditional and Western knowledge and science and use that as a part of the work that we're doing to help impact communities. So with that, I want to say thank you so much. This is our picture, our lab axe picture from Carissa's celebration, but all of this work would not be possible without my wonderful lab members who are really driving a lot of these projects, and their support means a lot to me. And also our tribal partners. It's been really fun to work with them. I was out on the Navajo Nation last week with a community advisory group, and then Leah and I are going to Montana in a couple weeks to work with their community group. So it's been really fun to finally be able to go out. For many years, because of COVID, we weren't able to go out to the community. So, and all of this funded by the NXURI. I can take any questions.
So what are some of the additional challenges that you face, like running like an interventional study and like more sensitive to other sort of populations like this versus just like an observational one?
Yeah, I guess I haven't run an intervention study yet, but I imagine that it will involve a lot of communication with the teams and making sure everything is culturally appropriate for those different sites. And then I think the Dakota sites and some of the Oklahoma tribes do have a cultural connection with smoking or tobacco in their ceremonies, but it'll be interesting with the Southwest tribe because we don't traditionally have a connection with tobacco. So there might be differences in how people perceive it. And then also we would just have to determine how, are we trying to, or we'd have to be very specific that we're talking about cigarette smoking, or I know a lot of tribal communities are interested in reducing vaping in younger adults. So I think just having all those conversations in advance would be the most challenging part.
Going back to the study where you look at beyond the Venezuelan genomic elements, I have two related questions. One is the variants that you find, is that consistent with other things that we know about those populations? For example, their food habits or their environmental conditions. And vice versa, what do those observing those genomic elements mean for us right now? How does it help us better understand human health in some way currently?
Yeah, and for your first question, you meant the Neanderthal populations or the modern human?
The archaic population.
Okay, yeah, so I don't know if folks noticed, well, there's only one high quality denisovan in the genome, so we weren't able to look at any more individuals, but with the Neanderthals, it's actually known that they're quite homozygous, so, and it was really good to see that if we saw one variant in one individual, it was actually reflected across most of the sites, so that was good for us to know, but they are from different time points and different locations in Europe. So, and in terms of looking at, or what are the implications for human health now, I think there's a whole field of evolutionary medicine that I'm interested in, and it relates to the variation we see today. How is it, or how did it come to be? So I think one of the variants that I'm really interested in is actually a hybridization, and we see the same hybridization in Neanderthal individuals as we do in human individuals. So it would be interesting to see if this was something that was introduced by introgression with Neanderthals, or was it sort of something that was separately evolved because there's a hotspot in that region or something. So I think there are a lot of implications. I think I'll have to think on that question a bit more, but I always like to know why do we see the variation we see today, and think about the cool interactions that could have occurred. Okay, I think we're going to switch to our next speaker. Thank you, everyone. Our next speaker is Maggie. I don't feel like I have to introduce you. I'm sure everyone knows you, but yeah.
Okay, can you actually use your
Sure. And thanks everyone for bearing with the video team.
Are you on Zoom? Maggie?
I am.
Can you don't have to plug in video, you can just share your screen.
Okay.
And if you want to turn on your camera, that makes it see.
Okay. I find the right. I always have so many desktops. Okay, and then on Zoom, is it like the right screen too? I feel like sometimes one is right and one is not. Is that okay? Click okay on it?
Seems good to me.
Okay, cool. So I'm going to be talking today about host genetics and gut microbiota in asthma. Oh, seems like we have a little thing going on with animations here, but so I think these like short talks were kind of supposed to, like introduce everybody to our like research programs and whatnot. I kind of gave that kind of talk at the DBMI retreat, and so I'm going to mostly talk about one specific project, but it's actually not my like main area of research, and so I wanted to mention a few projects that we have going on right now in my lab. So one project is Emily Yao, who's a PhD student at CU Boulder in the integrative physiology department. And she's working on a project looking at whether or not omic risk scores predict weight loss success during an intervention of daily caloric restriction versus intermittent fasting. So she's looking at genetics and DNA methylation and the microbiome. And she's presenting this work at Charge, if anyone is going to Charge. And then she also has an oral presentation at the American Society of Nutrition Conference. So if anybody is going to either of those, then check those out. And then Ashley, you guys know, she's here, is my research assistant, and she is looking at changes in the gut microbiome during this weight loss intervention, and then the relation to clinical outcomes. And so she is presenting that next week at the Front Range Microbiome Symposium. And then we also submitted to the Obesity Society, so we will see how that works out. And then this is Chris Earhart. He is working on a project that's kind of like an extension of a rotational project that he did with me and some other faculty. It was like a group rotation project. And so he is looking at polyomic risk scores of obesity in the multi-ethnic study of atherosclerosis, or MESA. And MESA is one of the like large epidemiological cohorts that I work with. And so that was kind of like the goal project, and that's one of his dissertation papers. But he kind of like went down a rabbit hole, if you know Chris, like he likes rabbit holes. And so he has done this like very extensive project using like genomic SEM to build like lots of obesity polygenic risk scores, like looking at all these like latent factors involved or related to obesity. And I was just saying, like, I think he was submitting it to Charge, so maybe he'll be there, but I haven't heard. So, but if anybody's going to those things, check them out. And you guys might know Chris Earhart. He used to work in Kathleen Barnes's lab, and he played a very fabulous role. It was very memorable as the Muppet Beaker in like a skit at one of the CCPM retreats. Do you people remember that? It's very good. Okay, so you might have noticed that none of those projects related to asthma. And asthma is not really a phenotype that I work on typically. But I got introduced to the Hispanic community health study of Latinos like a few years ago. And it's a really cool study. It's like 16,000 individuals. They're all Hispanic Latinos. They all live in the US. There's 4 different study centers across the US. And the individuals moved here primarily from Mexico, Cuba, Puerto Rico, and the Dominican Republic. And they're all like first or second generations, like living in the mainland US. And so there's a sub-study of SOUL that is about 3,000 individuals. And this is like a microbiome study. So this was the gut origins of Latino diabetes, or GOLD. And so they were aiming to examine the gut microbiome in relation to diabetes and obesity. And so when I found out about this sub-study and I was introduced to them, I was like, hey, I would love to be involved and like use your data. This is just like a really cool cohort. And I was like, I study mostly like obesity and diabetes. And they're like, well, sorry, no, like these are taken, they're like part of our main grant. And they're like, but we have asthma open as a phenotype. And I was like, well, I know nothing about asthma, but sure. So that's how I got here. So I applied for a biodata catalyst fellowship to look at like host genetics and the gut microbiota in relation to asthma in this like Hispanic Latino population. And for those of you who aren't familiar with the like biodata catalyst fellowship, I think it's now morphed into the Top Med fellowship, but it's a great fellowship to check out for like trainees and early career folks. It just funds like computationally intensive projects. So asthma, it turns out it's kind of a complex phenotype, which I learned, as I was applying for this grant and like doing this project. So it's one of the most common respiratory diseases in the world. But it's more of like an umbrella term. Like clinicians kind of think of this as like a lot of different phenotypes within the umbrella of asthma. So there's kind of like different clinical presentations, different like pathophysiological characteristics. And there's also really diverse risk factors. The environment like plays a big role. Things like diet and socioeconomic status and pollution have all been linked to asthma. And then there's also a lot of kind of interesting relationships with like birthplace and migration and race and ethnicity. And this is where like the SOUL study has really contributed to the literature and improved our understandings of some of these factors. So one thing that's really interesting about Hispanics and Latinos is that Puerto Ricans and Dominicans have a really, really, really high risk for asthma. And Mexicans and South Americans and Central Americans tend to have like notably like low risk for asthma. So across like Hispanic Latinos, there's like a really huge difference in prevalence. And this is like partly because of like genetic reasons, but potentially also because of environmental reasons. And also, this is not something I can delve into in this project, but there's also like complex interactions between all these things. So it kind of, you know, depends on like where you migrated from, but also like at what point in your life you migrated here to the US, but like there's different risk levels like in people's home country versus like, you know, Hispanic and Latinos who have moved here. And then the other thing that's also complex is that asthma is really highly polygenic. The genetic contribution tends to be like somewhat weak. So I think probably most people are familiar with AUC, but AUC is like a metric of like performance of like a model. And if its value is like 0, that means it's like totally not predictive of your outcome. If it's 1, and that's like perfect prediction. So 0.5 is like random guessing. And asthma polygenic risk scores are usually like 0.55 to 0.6. So slightly better than random guessing. Why does it go forward? Oh, there we go. Okay, so the other thing about asthma is that it's often comorbid with obesity. So obesity is both a major risk factor and a disease modifier for asthma in both children and adults. And one thing about obesity is that there's like a really extensive literature like connecting obesity to the gut microbiota. But you know, an open question is like, what is the role of the gut microbiota in asthma? We know that the gut microbiota interacts like really extensively with the immune system. So there's kind of like a mechanistic, you know, potentially mechanistic link. But there's not a ton of research in this area. And it's not necessarily like super obvious that like the gut would necessarily be involved in this like pulmonary condition. There have been more studies of like nasal, oral, and lung microbiota, and those have all been linked to asthma. And then a lot of the research in this area has focused on children. So like childhood asthma is kind of distinct from adult onset asthma. It's distinct like genetically, and a lot of the studies like focus on the gut microbiota have been in like, you know, infants and children. And then a lot of the like microbiota features and like taxa that have been linked to childhood asthma have also been linked to other like atopic disorders. So eczema, allergies, like those kinds of things. A lot of these studies have shown protective effects of short-chain fatty acids. These are, you know, they're from our diet, but they're also like made by the microbiome, and they're very powerful like anti-inflammatory molecules. And you know, asthma is like characterized by chronic inflammation, and so is obesity. And there has been like one kind of smallish study of asthma and obesity, and they saw like enhanced changes in the gut microbiota relative like to obesity alone or asthma alone. So this is the SOUL study. So there's about 16,000 people in the entire like SOUL study. The GOLD sub-study, which has like the microbiome samples, is about 3,000 individuals. We were also using genetic data from TOP-N, that was about 7,000 individuals. And then the subset that has like both types of data is about 1,000 individuals. So a lot of the analyses I'm going to be talking about is about 2,400 individuals, 191 of them have asthma. And this is just the overall study cohort. It's, you know, older adults, predominantly female, which is really common in cohort studies. Asthma is actually like more prevalent in females, particularly obese asthma. So like 84% of the individuals with obese asthma are female. And then this is showing the asthma and obesity prevalence. So this is what I mentioned before, that like asthma prevalence really varies across these Hispanic and Latino groups. So it's about 3% in Mexicans and close to 25% in Puerto Ricans. So it's a huge, huge difference. And there's also a huge difference in like the genetic admixture of these groups. So that's part of the reason. And then, you know, probably the environment, and some of it like we don't fully understand. And then obesity is like more common among people with asthma. And so like the blue lines here are showing like the prevalence of obesity like overall, and then the red lines are among like asthmatics specifically. So you can see that there is increased like prevalence of obesity in asthmatics, especially Dominicans. There was like 75% obesity prevalence. And then this is showing the kind of overall association between gut microbiota composition and asthma. So this is like a PC plot. So the dots that are really close together are like more similar in terms of their microbiota composition than dots that are like really far apart. And when we like analyze these statistically, like there is a pretty big difference between these asthma obesity like subtypes. We also look at alpha diversity a lot in microbiome studies, and this concept like comes from ecology. Like our gut is like a small scale ecosystem, but it's the same idea as like a large scale ecosystem. And you can see like in this picture on the left, like this is a diverse environment, and it seems like kind of overall healthy, like healthier compared to these places on the right. So high alpha diversity usually has like both high richness, like lots of different like plants and organisms, like in this picture, and also high evenness. And so here we have low richness, so like there's not a whole lot, at least at like the macro scale, not a whole lot of different organisms there. And then here we have low evenness, so we have like a lot of palm trees, but not much else. So those are both like low alpha diversity. And just like this picture on the left kind of looks like healthier. In the gut, that's also true. Like a lot of times, like a healthier gut has higher alpha diversity. And so we would expect low alpha diversity to maybe be associated with asthma. We don't see a strong association. Like, so we have 4 different metrics of alpha diversity here. And they kind of capture different aspects of like evenness and richness. And we did like sequential modeling. So like the model in black is kind of the one that's, like fully adjusted. And so we don't see like a strong association between alpha diversity and overall asthma, like when we group like obese and non-obese asthma together. But when we break it up into non-obese asthma and obese asthma, then we start to see a slightly different pattern. So like obese asthma is definitely associated, at least with some of these metrics, with lower alpha diversity. And then non-asthmatic obesity, like same pattern. But with non-obese asthma, we actually see like the opposite result with one of these metrics. And so there's, overall, it's like mostly being driven by obesity. We also looked at taxonomic associations with asthma and obesity. And I'm not gonna like go into too much depth about this, but we see that some taxa seem to like, there's current asthma in the like left, and then non-obese asthma, and then obese asthma, and then non-asthmatic obesity. And so we can see like the ones on the right are, these are like obesity, and then the ones on the left are more like kind of asthma driven. And you can see like some of these taxa are really kind of specific to obesity, and then some of them are kind of specific to both, or like more with asthma. And then we also did a Mendelian randomization analysis to look at, causality, and we saw that one of these taxa, pseudoflavinofractor, I think that's how you say it, was causally associated with obesity, and then this Fornariella was causally associated with, the first one was associated with asthma, and the second one with obesity. We also did a polygenic risk score for asthma, and we see like, these are violin plots, and we could see like the polygenic risk score is, elevated among people with asthma. And when we look at like non-obese asthma and obese asthma, with non-obese asthma, it's like a little bit elevated, but with obese asthma, it's like more notably elevated. So this is a polygenic risk score like for asthma, but it does better with like obese asthma. And we can see the same pattern with the AUCs. At the beginning, I mentioned like usually for asthma, the polygenic risk score AUCs are like 0.55 to 0.6. So these are actually like pretty good. But again, it's higher for obese asthma, so that 0.66 for obese asthma. We also did like sequential modeling to try and see like, okay, can we differentiate like asthmatics from non-asthmatics using like just kind of baseline characteristics, this asthma PRS, and then adding microbiota features. So this isn't necessarily like that useful when we do it cross-sectionally, which is what we're doing here. But a lot of times the hope with these risk scores, especially like the genetic risk scores, is that you can do this longitudinally so you can predict a phenotype before it shows up, right? So you can like predict somebody who is eventually going to get asthma or might be at elevated risk over the lifetime for asthma. And so we started with like a model of baseline risk factors, and then added the asthma polygenic risk score, and then added microbiota features. And for current asthma, so that's like non-obese and obese together, both of these, like each step increased our model performance. For non-obese asthma, interestingly, like the polygenic risk score actually didn't improve our model like above baseline risk factors. But the microbiota features did. And then for obese asthma, both were quite predictive. So another thing about asthma is that there are these like clinical subtypes that are like often seen. So one of them is younger, like childhood onset asthma. It's often comorbid with like allergies and atopic conditions. And then there's another phenotype that usually is like older onset, like adult onset, usually over 40. And it tends to be associated with obesity and with poor lung function. And so we wanted to know, do microbiota features like correlate with these asthma subtypes, or like can we see like distinct signatures? And the answer was no. So we built these like two subtypes, and the AUC was 0.52, which is really not very good, and like the confidence interval is 0.42 to 0.62. But what was kind of interesting is that the subtypes actually correlated really well with like childhood versus adult onset asthma. Like that was kind of the predominant like difference between the two. I mean, there's other differences as well, but like that was kind of a defining feature of these two phenotypes. And when we just like tried to predict childhood versus adult onset asthma, like the model actually did a lot better. So the AUC was like 0.59. And then the other thing that we wanted to do was like most of the comparisons I was presenting were comparing like people with like obese asthma to people with neither asthma or obesity, or like people with non-obese asthma to like people with neither condition. But we also wanted to see like, can we differentiate like these like asthma obesity like subtypes? So can we see a difference between like obese asthma and just obesity, like non-asthmatic obesity? And so we kind of compared all these like subgroups. And so this is showing like the AUC for those different models. And the best one is like non-obese asthma versus non-asthmatic obesity. So these are like kind of the most different phenotypes here. And so the AUC was 0.78, which is like actually pretty good. There's also a big difference between like current asthma, so like obese or non-obese versus like non-asthmatic obesity. So like those are pretty distinct. And then obese asthma versus non-asthmatic obesity, again, like fairly distinct. But then non-obese asthma versus obese asthma, not as different. So the AUC confidence interval does overlap with 0.5. So technically, like not really significant improvement. But there is also like a huge power difference. Like most of these comparisons, like we have a large group of people with obesity, but like non-obese asthma and obese asthma, those are like the two small subgroups. So more work should be done with larger samples. So some conclusions. Gut microbiota composition is associated with asthma and obesity subtypes. We saw that asthma was associated with altered gut microbiota. We saw that both obese and non-obese asthma have distinct microbiome signatures compared to non-asthmatic obesity. But like to try to differentiate like obese and non-obese asthma, like we probably need larger samples. We saw associations with alpha diversity. We did see a couple of taxa that had like causal associations using Mendelian randomization. And then we saw that the gut microbiota and the polygenic risk score independently improved models of asthma status, particularly for obese asthma. And interestingly, that polygenic risk score did not actually improve models of non-obese asthma. And then also the clinically defined asthma subtypes don't really correlate that well with microbiota. vital characteristics. So that's it. I just want to acknowledge everybody who's a collaborator on this, especially Liz Lakowski. Some of you probably know her. So she did a lot of work on Terra, Biodata Catalyst, which there was a lot of like slamming your head against the wall. So it was very nice and she was helpful with that. Thank you.
Yeah, so really interesting work, Maggie. And I apologize if I missed it. So kind of two questions here. One, was there a longitudinal nature to this, or is it all cross-sectional?
It's pretty much cross-sectional. So there's like, Seoul has 2 visits. And so we use data from both visits to define like, if they have a history of asthma or not. And so like we were doing current asthma, but like we excluded everybody who had a history of asthma.
Okay.
But the microbiome data and the asthma data were essentially at the same time.
And obesity.
What?
And obesity.
And obesity, yeah. They do have another study visit now, like that I think maybe just completed. And so there is like, and they're collecting microbiome samples again. So there is like possibility of more of like a longitudinal study.
Cool. And then this is an analysis on all of the Latinx samples together.
Well, not exactly. So a lot of, like I didn't go too much into the methods here, but so like for the beta diversity and the alpha diversity, that was like everybody all together. And then for the the taxonomic analyses, I split the data into like a testing and training set. So everything that I was showing was like in the testing set. So not all the data together. And then the other piece is like the national background groups. My overall analyses, I just like had everybody together. I did look at subgroups, and it's pretty hard to interpret because like the difference in sample size is there's just like a huge difference. But then also like the asthma prevalence is really different. So like I was really excited about that initially, and then we just like you couldn't really conclude a whole lot.
And is the microbiome different between those two groups?
Well, it is a little bit. I don't think that was like a huge determining factor though.
Okay.
Yeah.
Because I wonder if that would contribute to a difference in like predictive performance, or if you're not seeing right much.
Yeah, and I mean, definitely I'm kind of like, so the relationship between alpha diversity and every like disease kind of varies. But I think some of that is because like populations are really heterogeneous, and I've seen in other studies that like it seems like this association between like low alpha diversity and obesity is really mostly seen in white populations, and sometimes the opposite is seen in non-white populations, but it's really hard to look at because like we don't have very many non-white populations. But when because I'm interested in that, I looked in Seoul. And the groups that are like more white, so like Cubans in terms of genetic admixture, like Cubans have a lot of European ancestry. So they're more like quote unquote white. Mexicans have a lot of American ancestry, and then Puerto Ricans and Dominicans have a lot more African ancestry. And you could see like the same pattern that like the low alpha diversity association with obesity was like much stronger in Cubans and Mexicans, and that you really couldn't see it in Puerto Ricans and Dominicans. So I think all of that is interesting, but again, it's like hard, like we only had 190 cases of asthma, so it's like to break things down more and more and more, it's like, at a certain point, you can't do it.
That's really hard. And also, I mean, from my understanding, there's not as much of an association between microbiome and ancestry versus microbiome and like culture and ethnicity. So kind of trying to disentangle.
Yeah, and that's definitely something I'm very interested in trying to disentangle. But again, we need like more diverse, like large samples to do that. But yeah, I mean, like you could see how like genetic admixture, like that's a reflection of your like evolutionary history and like where you came and like the diet that you like evolved with, like that's gonna be reflected in your microbes. But then like what you self-identify as, like that reflects your current diet, it reflects your current environment, like, in this country, like your socioeconomic status, like all of these other things that are like, distinct, but also really important for the microbiome.
I'm really interested, Maggie. This is great to see. Nearing completion. I'm really curious about the asthma PRS. Like, I think it makes sense, I guess, because they're comorbid, that they have shared genetic risk. Do you have a sense of whether an obesity PRS, like, similarly would discriminate those groups, or like, what is the shared
Yeah, so
genetic?
that is definitely something I was glad a reviewer didn't ask, but I was interested in it.
I was not a reviewer.
Yeah. Yeah, no, I was interested in that, so it was kind of like, oh, I would like to do that, but like at a certain point we have to like stop, right? You have to stop project. But it has actually been done in Seoul. So like they looked at obesity PRS with asthma and it was predictive. And I think it was more predictive, I want to say, of childhood asthma than adult onset asthma. And then we did look at the genetic correlation. I don't think that's in the current version of the manuscript, but we did look at the genetic correlation. It's not like overwhelming, but it's definitely significant. There is some pleiotropy. Like this has, you know, been studied in other studies. Like it is known that there's some like overlapping risk factors. Yeah.
You said that you had problems, or like that your microbiome models weren't discriminatory against like your clinical subtypes of asthma.
Right. That
So would you mind describing like how you were building these models? Really, just like ASV vectors or?
Yeah, so we used like, and this is like something also that we like took out of the manuscript now. So we built these like clinical subtypes. So we used, I think I want to say like unsupervised learning, like based on these clinical, all this clinical data. Not all of it was like in our control models either. It's like COPD status, and you know, medications, like we had all sorts of like clinical risk factors. And so then we like clustered the two groups, and then we looked at like, okay, so what are the characteristics of these two clusters? And it worked out that like we saw the two clinical clusters that are kind of consistent across the literature. And also if you talk to a clinician, they'll say like, we have this childhood asthma group, and then we have this, you know, like mostly female, over 40, adult onset, whatever. So like those are the two groups that we got based on like our unsupervised learning. So that was great, like step one was great. And then we tried to see if there was any microbiome like signature between those. And so I think what we did in the end was like a lasso model to try and like predict them and then see how well it performed, and it just wasn't good.
Okay, so I guess you just like your microbiome features are just like your ASVs then, right?
Yeah, so I think, yeah, we might have used species level, and then we also, I think, used alpha diversity, like threw that in there too, and like just not great.
So do you think that if you use like some like incorporated like phylogenetic information, like an Adonis test, like you've got your unifrac distance matrix,
Yeah.
and basically use that to try and predict like your clinical stuff.
So just put that into Adonis?
Yes.
That wasn't significant either.
That wasn't significant.
Yeah. Yeah, but again, like, so this is the subset who have asthma, right? Like this is subtypes of asthma. So we have 191 people, which when you think about like the microbiome's heterogeneous, we have these like national background groups, probably different diet, like all this stuff, like that actually probably isn't a huge sample size. So I would say like the jury's not totally out, like maybe it would still be useful for that, but I think you need like 1,000 people or more.
I just kind of shoot from the hip here, but I'm guessing there's probably like more interpersonal variation in like respiratory microbiomes.
Yeah.
Then I guess like what you have in your gut, just kind of like hillbilly reasoning. There's just like more gene flow coming in and out of the environment. You can have like more transient things hanging around long.
Yeah, that's probably true.
Thank you.
Cool. Thank you.